In this video, we're going to look at how a colorimeter works because we will be using one in an upcoming lab. And I have in parentheses the word spectrophotometer. In our lab, we have colorimeters and spectrophotometer. They're a simpler version of something called a spectrophotometer. So in a spectrophotometer or a colorimeter, there's basically three parts. There's first a light source, and that's the biggest difference between a colorimeter and a spectrophotometer. And a spectrophotometer, the light source, wavelength of light, can be varied through all the ranges of wavelengths of visible light. So in a spectrophotometer, you get the entire spectrum of visible light, and you can look at your sample through any color. In our colorimeters, we're limited to, in our specific case, we just have 4 specific wavelengths of color, and we pick one of those 4 to analyze our sample. We can't look at the whole light spectrum. We have a light source. That light source is shown through a colored sample. I made ours purple because in this lab we're going to use crystal violet, which you'll see is a very deep purple color. You pick a wavelength of light that when it hits the sample, some of that light will be absorbed by the sample. Usually you want to pick a color that's opposite the color of your sample on the color wheel, because if your sample appears purple, it's because it's absorbing colors on the opposite side of the spectrum, and all you see are the purple types of wavelengths. So we're going to shine a light source at our sample. Some of the light will be absorbed by the purple sample. Some of the wavelengths of light will be reflected and pass through, and those wavelengths that go through the sample will be detected by a detector, and we will be able to measure. The detector will tell us the absorbance of the sample, and will tell us the absorbance of the sample by comparing the light that comes from the light source to the intensity of the light that hits the detector, and that will tell us how much of the light is actually being absorbed by the sample. In order to demonstrate the variables that have an effect on the absorbance of light in a colorimeter, we're going to look at a couple of samples of food coloring. And in these samples, we're going to look at them in 2 ways. First, I want you to compare these 2 beakers. The beakers currently contain solution of food coloring in water with the same concentration, and each beaker contains about the same volume. First, I want you to look at the beakers themselves. As you look at the beakers straight on, you're basically looking this direction through the liquid. So we've got the same concentration, and we have what's called the same path length. Basically the distance, the width of the solution that you're looking through. And if you look at those 2 beakers, they should appear to be approximately the same color. Next, I want you to look at the overhead projector image of the 2 beakers up on the screen, so up here. Notice again, this blue and this blue look to be about the same color. This time, the way that we're looking through the liquid is, so this time as we're looking, the light from the overhead projector is shining up from below the sample. It's going through the sample, and then what's projected on the screen. This time, because we're looking at something, these images have been created by looking in this direction, but because we have the same volume of sample, the height of the water is the same. We again have the same concentration and the same path length, so the color intensity looks basically the same. Now we're going to change some of those variables and see how that affects the deepness, the intensity of the color that we see. Next, we're going to look at 2 samples. What I did in this scenario is I poured more of the same solution with the same concentration into the beaker that has the red car sitting next to it. And I put the car next to it because on the overhead projector, you'll see that the location of the beakers is kind of crisscross. So the image on the overhead projector over here is the image of this beaker, and the image on the overhead projector here is from this beaker. So first, I want you to look at them straight on in this picture. So our path length is from your eye through the solution and then out again. So the path length is the same on both of these, and the concentration is the same. So if you look here and here, the deepness or the intensity of the color should look about the same. In the second picture, I want to focus on the images that are made on the overhead projector. So notice This time on the overhead projector, this sample appears darker, and notice it's the one that has the car by it. You can tell by the little shadow from the car. So it corresponds to this beaker here. They have the same concentration. I just poured more of the solution that I had made into the beaker with more volume. In this case, the light from the overhead projector is originating below. It's going up through the solution, and the image on the overhead or on the screen is being made but because of light traveling up through the solution. So in this case, the beaker that has more liquid in it has a longer path length because the light traveling through the liquid travels through more of the solution. And so it seems like doubling the path length by doubling the height of the water results in a more intense color of the image that comes out the other side. For the last set of comparisons, I took the original two beakers that we had that each had 100 milliliters of solution in them, and I added water to the second beaker to dilute it to twice the volume. So now the concentration of the original beaker, I've got another beaker with a concentration that's half of what the original concentration of the solution was. So I've changed the concentration of the solution. In this first set of pictures where we're looking at the beakers straight on, we've got the same path length because in both cases we're looking through the width of the beaker, the diameter of the beaker. But the concentration is different, so notice that the second beaker looks lighter because the concentration is lower. So here we're seeing that concentration has a direct effect on the color that we see. Now on the other hand, if we can see the effect of path length when we look at the view of the light shining up from the bottom of the samples, going all the way through the sample and then being projected on the screen, notice in this case the color of the blue looks about the same, even though when we looked at these beakers straight on, the color was different. Here, what's happened is we have doubled the path length in the second picture, but we've halved the concentration. And since those two effects kind of cancel each other out, doubling times 1/2 leads to no change in the color. They kind of counteract each other. So there's no net change in the color intensity. So it looks like from our investigation that making a longer path length will make the color intensity bigger, and making a concentration darker will make the color intensity bigger. So that leads us to a relationship that is employed when using colorimeters. So let's look at it on the next page. The relationships that we've been looking at on the last few slides are summarized in something known as Beer's law. Beer's law is a way of it's looking at how the absorption of light by a sample depends on some other variables. So A stands for the absorption. Then we've just investigated two variables that both show up in this equation. B stands for path length. Don't ask me why they chose the letter B. I am not sure, but I can tell you that this equation is on the AP reference sheet. And in case you forget what each of these letters mean, because we don't use this equation a whole heck of a lot, it does define right next to the equation that B, for example, equals path length. C stands for concentration, the two variables that we just investigated. And then the script E stands for something called the absorptivity. And absorptivity has to do with, or it's the absorptivity of the solution. And as long as you've got the same chemical, like we're going to do a lab all with crystal violet, it's a constant. So this mathematical expression can explain all the things that we just witnessed. For example, we saw that if we took the concentration and we doubled the concentration, that would make the color that we saw twice as deep. We also saw that if we doubled the path length with the same concentration, that would make the absorption twice as dark, or the color would be twice as deep. And in the third example that we looked at, we saw that when we doubled the path length but halved the concentration, that there was no change in the color, that the two samples had the same color. All of this talk about spectrophotometers or colorimeters and the relationship between the absorption of light and the concentration of the solution all become very helpful, because when we want to monitor a reaction that involves a change in color as time goes on, and we want to monitor how quickly that reaction occurs, we can use a colorimeter or a spectrophotometer to monitor that in real time, which means we don't have to stop the reaction every so often and take a sample and check how it's going. We can just monitor the color, use the color to infer how the concentration is changing, and determine and study the rates of reaction. That's exactly what we are going to use this for in our lab this week.